This is episode 219 of the Stem Cell Podcast, Using Innovation to Change Lives, with Dr. Stephen Stice. Hey, everybody, this is Dr. Daylon James and Dr. Arun Sharma. Welcome back to the Stem Cell Podcast, where we culture knowledge in stem cell research by talking to some of the brightest minds in the field. The Stem Cell Podcast is brought to you by STEMCELL Technologies, a global biotechnology company supporting life science research and fostering communication and collaboration in science. Today, we have Dr. Stephen Stice from the University of Georgia. He's on the podcast to talk about his research on developing novel therapies and new technologies for drug screening, neurodegenerative diseases, animal agriculture, and human biomedicine. We've also got our usual roundup of recent highlights in stem cell news that's coming right up.
But first, neuroscientists looking for more predictive power in their disease models are increasingly adopting human pluripotent stem cells in their research. STEMCELL Technologies offers products, protocols, and training to support HPSC-derived neural models. Explore their collection of technical videos and webinars on neurological disease modeling by visiting www.stemcell.com/neural disease model.
Arun, I'm starting the roundup today by talking about something that plagues us all, aging. We're not getting any younger, and aging in itself as a, condition, I wouldn't call it a disease. Better to be aging than to be dead, right? It's manifested by waning regenerative potential, and that results in declining tissue and organ function. But amazingly, very promising technology for us vampires out there. There have been some reversal of these aging phenotypes in animal models, at least, and in vampires historically. Most notably, as I keep referencing, through the use of this heterochronic parabiosis, right? And that's the system that they use in mice, maybe other animals too. I don't know if they've done that in other animals, but in mice, they do it all the time. They hook up the circulatory system of aged and young mice surgically. So they're connected, and you get this, sharing of these blood-borne factors. And what's amazing here is that the young blood is capable of reversing a lot of age-related structural erosion, molecular changes in the muscle, bone, liver, and nervous systems. And that's just what, we've looked at and found so far. And although there have been a lot of putative pro-regenerative, pro-youth factors that have been identified, I think, some of them are still controversial and debated. But the factors themselves, at least the candidates, have been identified. But the understanding of their targets, the downstream tissues that are affected and rejuvenated, and at what level, is not really well understood at all. So when we talk about aging, of course, one thing to think about, and that's why we do what we do in our research and on this show, is stem cells, right? The systemic aging is attributed in large part to the impairment and exhaustion of these adult stem cells, organ-specific, tissue-specific, although they may be. And because these resident adult stem cells are compromised by aging, it's thought that, they're a prime candidate for what these blood-borne factors are targeting. But whether or not that's true and to what extent it's true is not understood. So in this study, we have a pretty big group here from a Chinese Academy of Sciences, the Institute for Stem Cell and Regeneration, which was led by Jing Ku, Wei-Qi Zhang, and Guanghui Lu. Also there, the lead contact, interestingly enough, because he's everywhere, is Juan Carlos Espizua Belmonte, who's not at the Chinese Academy of Sciences, but he always has his hands in studies like this. And what they did as a group there, and a lot of work, a real tremendous amount of work, is they systematically analyzed the single cell transcriptomes across the hematopoietic and related immune system, which is the hematopoietic progenitors, bone marrow cells, peripheral blood, some extramedullary sites like the spleen here. They also looked at 4 solid tissues / organs: the skin, skeletal muscle, brain, and liver. And what they found, as you would expect, of course, is that heterochronic parabiosis rejuvenated the adult stem cells across all the tissues and their niches, I should mention. But really, in particular, and this is the great insight, is that the parabiosis really rejuvenated hematopoietic stem and progenitor cells. They were the most responsive to this exposure to young blood. And there was a continuum of changes across hematopoietic and immune cells. And what it ultimately resulted in is a restoration of this youthful, quote unquote, transcriptional regulatory program and restoration of the expression of all those cytokines. Um, and interestingly here, this was, I think, what maybe got it up into cell stem cell. Although, I mean, if I'm being perfectly honest, I didn't think they went really deep on this. They were able to show that if you reintroduce, quote unquote, some of the identified rejuvenating factors alone, they were able to alleviate, in this case, the lymphopoiesis decline. Okay, so a very specific phenotype. If you kind of hack the system. Although technically, I wouldn't say that they introduced a factor as much as they knocked out a factor in the context of parabiosis and showed that the parabiosis was no longer as effective. So this isn't yet kind of the secret sauce or the magic bullet that we can just give people a cocktail and rejuvenate their niches. But I think for sure there's a really great resource. I mean, this is a cell stem cell resource here because it gives you a really comprehensive data set with a lot of cells and a lot of information from a lot of tissues, indicating how perhaps this heterochronic parabiosis mediates its effect and showing some of the downstream signaling pathways that may underlie that rejuvenation. So a great resource, fertile ground for future study. And I mean, a huge data set, to be honest. And we always have been interested in this, it's almost mythical, this idea of parabiosis and rejuvenation. So it's nice to see some science come in and support it.
Yeah, anytime you talk about parabiosis, it captures the public's imagination. It's one of those areas of science that's really caught on, and for obvious reason, right? You're thinking about the idea of rejuvenation, as you mentioned, and undoing aging, and all these exciting things that have a lot of startups, a lot of money being pumped into this. We talk a lot about the different startups that are focused in this particular area, even some stem cell focused startups. um, but yeah, I mean, this is, like you said, this is, I think, first and foremost, a powerful resource. It's a stem cell resource for a reason, right? The data it's a great data set that you can utilize for other folks who are doing work in parabiosis and looking at, in particular, the impacts of parabiosis on these secreted factors in the blood. Um, I think there are a couple of things to consider here. One, the case with any single cell story, and we're going to talk about more single cell stories in the next couple papers in the next segment in the roundup too. Case with any single cell story is the limitation of the cell number, right? You have to have the appropriate number of cells to actually make some of these conclusions when it comes to the secreted factors, and ultimately utilizing some of these secreted factors to reverse aging and do some more in-depth mechanistic studies. Okay, so that's one. And they actually directly mentioned that, we have to get more tissues, more cells. And that's the case throughout single cell biology. I think the other thing to consider here, and this is something to do with more of the technique of parabiosis itself, right? What are you doing here? You're actually sewing together two animals, and that in itself is an inherently stressful process. Okay, would that impact the secreted factors in the blood? Absolutely. I think that that's definitely a variable, a caveat to consider. It's not exactly the same as doing, say, a blood transfusion from young to old and seeing what the changes in the transcriptome is there. It's a stressful process on these mice. Anytime you're sewing two animals together in the form of parabiosis, that's going to impact the downstream transcriptional phenotypes, don't you think?
Yeah, for sure. I would imagine that's a pretty aggressive intervention. But that last part that you touched on, I think, is interesting to me. It's not the same as a transfusion, right? Um, and that's the question for me that I think next needs to be answered. Maybe it already has, and I'm just not looking deep enough. But like, do you need to sew these animals together? And I would expect that you probably do. It's like a continuous um, circulation of these factors that mediates effect. Otherwise, maybe it would be enough just to transfuse these older mice. And that leads me to the kind of elephant in the room, medically, scientifically, is I, I'm kind of surprised that there aren't some kind of, you know, amongst the wealthy elite, just harvesting young blood and transfusing it. I mean, it seems like something that people do a lot weirder stuff amongst those wealthy elite. Believe me, there's a lot of NAD infusions and people are taking testosterone. I don't know why somebody's not hooked up to an IV with some young blood, or maybe they are.
You're going down a very dark road here, Daylon. This is definitely not something we are encouraging, but I will tell you, it's stuff that's probably happening in the dark alleys and the dark sections of the world. I mean, there are, like I mentioned, this is a hot topic anytime you're talking about aging and reversing aging. So I'll leave it to our listeners to do their own research here, but that's all I'm going to say about that. So moving on to another kind of single cell focused set of studies here, set of story. This is big news. Okay, this is really big news. We're talking about doing single cell and parabiosis and heterochronic parabiosis, but we're going to take a step back and really talk about these 4 huge papers in science that came out as part of the Human Cell Atlas consortium that a lot of us have heard about. And this is an international effort which has basically done single cell in a variety of different tissue types in the body, in the human body. And ultimately, what they want to do is, quote unquote, form a Google Maps, all right, a Google Maps of the human body so that you can look at any sort of gene in any tissue with some caveats, as I'm sure Daylon's going to mention here. Um, and basically see where your favorite gene is expressed. We've all had that question, right? We've had a gene of interest and we're like, oh, where is this gene expressed? Is it only expressed in my favorite tissue type? And this is a data set. And one of the papers here is really a great data set for help you answer that question, that exact question. Okay. So these 4 studies were simultaneously published in Science a few weeks ago. They're international teams of researchers that are offering, different single nucleus and single cell transcriptome maps of more than 1 million different cells, human cells, importantly, across 5 different cell types in 33 different tissues and organs. So just a tremendous amount of work here. And they uncovered some interesting features, right? Looking at different signaling mechanisms, genetic traits. So I'll, start off by just mentioning the names of the 4 papers. Okay, so the first one is I think the real highlight, the crown jewel of these 4 papers. This is the Tabula Sapiens, which is this multi-organ single cell transcriptomic atlas of humans. And I was actually playing around with this a little bit before the show, and it's a cool online data set. Anybody can access it. You can look at basically this UMAP, this scatter plot, this cluster plot of different cell types, different tissue types, and see where your favorite gene is expressed. Okay, that's pretty straightforward. The next one is another single nucleus cross tissue molecular reference map. This is single nucleus. And I think for the important clarification here is some cell types, some tissue types are more amenable to single nucleus RNA sequencing as opposed to standard single cell sequencing, because, some cells like larger muscle cells and all that, they don't tolerate single cell sequencing as well. These are just bigger cell types that just don't go through that process as well. So dissecting down to the single nucleus can help get around some of those technical hurdles. Okay. Then we have a couple of immune focused papers. Cross tissue immune cell analysis reveals tissue specific features in humans. And then a developmental analog of that immune paper, which is mapping the developing human immune system across organs. So these are 4 titanic papers out there in science that are really focused on single cell, all things single cell, international collaborations between the Wellcome Sanger Institute, University of Cambridge, folks here in the States as well. And so, what do they find, right? So one thing, for example, is they tracked blood and progenitor cells in immune tissues and also in embryonic gut and skin. And they found out some beta cell progenitors receive input from their development from immune cells from the gut, in addition to the bone marrow. So this is going back to the idea of the niches and the importance of different bodily niches in specifying cell development. We've actually talked about this a little bit on the show. Okay. So this is important because it can maybe influence how you engineer immune cells out of the body, like all these different immunotherapies that we're talking about. Maybe these single cell maps can help develop better immunotherapies. That's one application. So in the second study, when you were looking at the immune cells in particular, they developed this machine learning algorithm called cell typist to actually automate cell type identification, and they created this immune cell atlas, right, that reveals this relationship between the immune cells in different tissues. Again, one hope is maybe we can use this atlas as a way to develop better cell therapies down the road. Also, I got to mention one of the, of course, one of the titans involved in this particular project is none other than Aviv Regev, who's actually recently moved on to Genentech to take a position as a VP of research. But she is really the queen of single cell, as we'd like to say. And in the other study, they're looking at the tabula sapiens, right, which is like I mentioned, the real crown jewel here, led by Steve Quake over at Stanford, 500,000 different cells from 24 different tissues and organs, really a focus on collecting samples from the same donors, which allowed researchers to kind of screen out different background variations such as age. But it's just, I can only say so much. You really just have to look at the data set. You have to look at the data set, take a deep dive into it, look at your favorite tissue, your favorite gene, see where things are expressed, and just play around. And one last thing here, specifically going down to more mechanistic approaches as opposed to, oh, we have just a bunch of data, a bunch of cells to show you. They also showed that the CD47 protein, which some different teams at Stanford, like Irv Weissman's group, for example, they've shown that it's implicated in cancer. It's in the buildup of different arteries, different plaques in the artery walls. CD47 is actually being investigated as an anti-cancer therapeutic pioneered by Irv's lab. Here, they're actually showing that the CD47 could have this dual impact, this dual effect, not only in cancer, but also the buildup of the plaques on the arterial walls, again, based on the single cell profiling. Okay. So all different sorts of things, a few caveats, as I'm sure you'll talk about, Dalen, but like we're talking about, this is just a huge data set, great roadmap, ideally a Google Maps of the human body. And down the road, certainly there's going to be different versions of this. This isn't the end all. This isn't the end of this particular human cell atlas. They're going to keep on coming out with different versions of this, more cells, more tissues, but fantastic start and a great resource for the community.
Yes, amazing resource, a lot of fun just to play around with. And I'm going to tap this resource. I can't even tell you how many times, a lot. But you said it. I don't know about a caveat, but I will say when you talk about Google Maps of the body, I mean, last I checked, we had gonads, male and female, and even some of us that don't identify. Sometimes we got gonads, most of the time. No testis, no ovary in this data set. I was disappointed to see that. And I'm not trying to throw shade on this. Like I said, I'm going to use this resource. And I think that's the best testament to the utility of the thing is whether or not it's used. So it's a win already. I've already been playing with it. But I will say, I think that there's this, I guess they're trying to draw a parallel with like Human Genome Project, maybe. I don't know. That's the first thing I think of is this monumental effort to do something that'll kick open the door. And I just don't think we're at that level. I don't think that really it meets the need that's unmet. I like the effort to try and consolidate all the cells and tissues from a single donor. I think that's a really critical thing. Similar to the Human Genome Project, you need like a single reference, and then you can start looking at heterogeneity between people. But if you don't have it from a single person, then I would argue that maybe what you have out there is an aggregate of all the single cell data sets that are already there. Maybe it would be close to or approximate the utility of this thing if we just went on Geo, took every 10x chromium data set we could find and threw it together in one merged library. I don't know, maybe not, and probably not. I'll grant you that, probably not. But I just think that that's my caveat there is that there's a little bit of redundancy to it. And it's also not totally inclusive. So I think it falls a bit short on both ends there, which I think are minor caveats, if I'm being honest and fair, because it is such a monumental effort. And to have it all done by one, you know, collection, I think is key because you're not dealing with all these other variables. To have like a very robust QC and normalization process, which I presume they did have, because it's tough when you're looking at highly variable expression levels of a gene between tissues. You know, something might be expressed bonkers in one place, and relatively, you know, 5% of that in another tissue, but that 5% could also be critical in that other niche. So I think that there's a lot technically that had to go into leveling this data set so that all the cells could be looked at in one place. And that to me is the major accomplishment here, is to see all these cells in one map is a tremendous resource, and it will be used.
That's the key. I agree with you entirely. It's a resource for the community. It's a free resource. It's easy to use. Anybody can pull it up around the world. I've got it pulled up right now on the other half of my screen. It's literally they simplify things. Even if you don't have experience with Syrah or any of these complex single cell approaches that take a little bit of expertise and knowledge in R and stuff like that to use, you don't have to have that, right? All you need to do is to look at this plot, type in your favorite gene, type in your favorite tissue, and bam, it's there in a couple seconds. That's powerful. And I think another part of this is education, right? Bringing this kind of data set is not only useful for the advanced scientific community and like PhD level researchers like us, but potentially even, students could use it. College students, high school students, who knows? Like they're learning about genes too. And one of the most simple things of human biology is, figuring out where a certain gene is found and how certain genes are being expressed in the body. So I think this is it's a tremendous resource. Caveats withstanding, as you mentioned, the lack of the ovarian tissues and reproductive tissues. Where's the reproductive single cell atlas, Dalen? Are you working on that? Is that going to come out next week?
I
Tell me
better
about
get to
that.
work. Better get there because not a lot of respect for the reproductive biology out there in the stem cell field, it seems. But we'll get there. We'll get there, Arun. And again, to emphasize the tremendous value, this is a resource. It tells you everything you need to know about normal, right? About tissue in a physiological state. And that's my segue here to our last Roundup paper, which is looking at pathophysiology, in this case of the mammary stem cells and resultant cancer. And it's a story about X inactivation at that. Wow, a hodgepodge. So yeah, X inactivation, right? It's the transcriptional silencing of one of the two X chromosomes in female cells, right? And the way this works is that the X inactive specific transcript, also known as EXIST, is upregulated in early development, right? Very early, from one of the two X chromosomes, and that coats that chromosome in the EXIST and leads to a cascade of epigenetic events that impose this compacted chromatin state, or silenced state. But EXIST, it remains highly expressed in cells throughout life and remains tethered to that inactive X chromosome. But the question has been, do you need the EXIST? Clearly developmentally, you need the EXIST there to coat the chromosome in the first place and initiate this inactivation. But does it need to stay there to maintain the inactivation? And that's still debated. For a long time, it was thought that it was dispensable. You could get rid of EXIST after the fact in somatic cells. And there were studies showing that you delete it and the heterochromatic inactivated chromatin, it remains. The X chromosome remains inactivated even after deletion of EXIST. But there have been other studies that used a more, I guess, comprehensive set of genes that had a wider view and more sensitive assay that showed that there can be transcriptional reactivation when you delete EXIST, right? But does this have disease relevance or relevance to biology in somatic cells in adulthood? That question remains open, but there's some evidence to suggest that it does, right? In mice, when you knock out exist, these mice present with impaired maturation of blood lineages, and they get these hematological tumors specifically in females. Okay, so that suggests that exist actually is doing something to tamp down expression or to regulate expression in the somatic cells throughout lifespan. And another thing here in the humans, it's been shown that there's aberrant expression of exist in breast tumors, right? So all this data together suggests that at least in some context, that maybe the reactivation of these X-linked genes can interfere with cell homeostasis and even elicit carcinogenesis or tumorigenesis. So that leads to this study, which was performed by Christophe Guinestier, who's at the INSERM in Paris, and also Raphael Magueron, who's at the Sorbonne. I'm trying to work on my name pronunciation there, guys, if you didn't notice. Doing my best. My apologies. I know I didn't do it right, but I'm trying. Anyway, those two gentlemen and their group, they set out to answer this question, whether or not exist function in the somatic cells is relevant to breast tumors. And what they showed by knocking down exist in mammary stem cells in human mammary stem cells is they showed that, yeah, indeed, you knock out exist and you get these highly tumorigenic and metastatic carcinomas, right? And that's because with this exist deficiency reactivate genes, a bunch of genes, but they followed up mechanistically on one of these, which is called mediator subunit MED14. We'll call it MED14. And what they find is that MED14 reactivation and overdose leads to hyperactivation of this transcriptional program in mammary stem cells that makes them less likely to differentiate. And then furthermore, so that's drawing a line between the loss of exist and the behavior of the cells. And then they show that the loss of exist and reactivation of the X chromosome is actually pretty common amongst human breast tumors with a poor prognosis. So I think all told, this is a really nice study that goes, it's got a little bit of organoids, some stem cells in there, some CRISPR, it's got all the pieces, but for me, it's really a nice demonstration of one, the exists relevance to disease in adults, and specifically in cancer, and really interestingly in this kind of gender specific fashion. I think it's fascinating, the idea that there may be a whole range of diseases or conditions or whatever, cell behaviors in women that may follow from this, pathological or whatever you want to call it, maybe benign reactivation of the X chromosome. So I think a nice little conceptual query there that's got me thinking.
Yeah, I agree with you. It's something that I guess we haven't really thought about, or at least I haven't thought about too much, this idea of gender specific malignancies and cancers, right? X inactivation, X reactivation is something that's exclusive to, women, and that's a unique thing that could happen in other tissue types as well, not only just breast tissue. So it'll be cool to see extending these studies beyond into other tissue types, see if there's other forms of malignancy based on X-mediated reactivation. The other thing to consider here is it's not just, there's sort of like an internal control. That's kind of what I'm going at here. There's sort of a built-in internal control here because men also develop breast cancer. That is one thing to consider here. And obviously, you can't really have this X-mediated inactivation or reactivation in men, in males. So that's perhaps something they could allude to or refer to in the future. But yeah, I agree with you. I think this is a really interesting concept for them to look into in the future.
Absolutely. And the male female thing, I mean, they did say it's really specifically noted in breast cancer, poor prognosis. So exist dysfunction is not really a hallmark of all breast cancer. But to your point about the male female, what was really fascinating to me, if you just look at the numbers, uh, cancer incidence, all type, is actually 2 to 1 male to female. So two out of three males versus 1 out of 3 females will get cancer, and non-reproductive, if you exclude reproductive organs like breast. Um, so that's a really interesting idea of it in itself. And there was work done about 4 or 5 years ago now at Dana-Farber by Andrew Lane that looked into this. And the idea was that maybe males were exposed to, riskier lifestyle or increased carcinogenic exposure, whatever. But what they actually found, which was fascinating, is that on the X chromosome of women, there are actually some tumor suppressors that retain expression, that continue to
Hmm,
be expressed, even on the inactive chromosome. So
cool.
it's interesting here. You have this interplay where here we're talking about, women maybe at increased risk because of exist dysfunction, but inherently it seems like they may have a greater resistance to cancer because of these tumor suppressors that remain active on the X chromosome. So as the information continues to emerge, the story just gets more and more complicated. It's difficult to predict, I think, 1 to 1 how who's going to be at risk for these conditions, cancer, namely, just because there's so many factors that play into it. But here's another, I think, to consider, and something, as you said, that maybe we should consider outside of just the breast and other tissues and organs that may be differentially affected in women due to this uh, duality of X chromosome inactivation. So I love it. I love this story. Came out of Cell, strong group from the INSERM and the Sorbonne. With that, we are finished with the roundup and we'll push on to the interview. But before we get there, I have a quick message from STEMCELL Technologies, who's conducted a survey asking scientists to help highlight the hurdles to genome editing using CRISPR. You can read that survey report to learn about the most interesting insights on topics such as editing efficiency and downstream viability and how to overcome them in your research. Visit www.stemcell.com/CRISPR survey results to learn more about that. All right, everyone. For this episode of the Stem Cell Podcast, we have joining us Dr. Steven Stice, who is the D.W. Brooks Distinguished Professor and Director of the Regenerative Bioscience Center at University of Georgia, also co-founder and Chief Scientific Officer of Aruna Biomedical. Dr. Stice is a world-renowned expert in the field of pluripotent stem cell biology. His group produced the first cloned rabbit in 1987. That's like 100 years ago now. The first cloned transgenic calves in 98, and the first genetically modified pluripotent stem cell derived pigs and cattle in 97. In 2001, he directed work on the derivation of 3 human pluripotent stem cell lines, which were approved for federal funding by the NIH and President Bush. It's a tough nut to crack. He has over 30 years of research and development experience in biotechnology and is a co-founder of several biotech startups, including Aruna, as we said, a company using neural-derived exosomes to treat neurodegenerative diseases. Currently, the Stice Lab is developing novel therapies and new technologies for drug screening and neurodegenerative diseases, as well as developing livestock technology to address the needs of animal agriculture and human biomedicine. Dr. Stice, thank you so much for joining us on the show today.
Well, thank you for having me. Pleasure to participate in this podcast.
Well, it's really an honor and a privilege to have you here. Let's go back to the beginning. It wasn't exactly 100 years ago, but it's, in stem cell time, it was kind of before stem cells. It's prehistory almost here. Back in 88, you demonstrated that individual blastomeres from an 8-cell stage rabbit embryo could undergo nuclear reprogramming and generate clones. Now, this is a major contribution to the long road leading to cloning from an adult cell, Dolly, and ultimately, I think, to the concept and application of induced pluripotency. And young scientists really need to recognize how unlikely this whole concept of induced pluripotency seemed back then. Did you imagine that reprogramming of blastomeres could be extended to adult cells, and eventually maybe to the complete chemical reprogramming of adult cells that we just reported a couple of episodes ago that was in Nature journal? That's a long way from, blastomeres and cloning, going to complete chemical reprogramming. Did you ever imagine that we would reach this point?
Oh, it was always a dream that this would happen. Nobody thought back then that you could take just a few reprogramming factors when Yamanaka initially made the discovery that, it would be that simple. And isn't it true that most things that are true breakthroughs are really simple solutions to a very complex problem? So we were, very excited about cloning rabbits back when I was in graduate school. Rabbits, they're really interesting. And why would you clone a rabbit? I often ask that of audiences. Why would you clone a rabbit? And I get a lot of blank stares coming back at me. But there really wasn't a great idea or a reason for cloning rabbits, other than mice were so difficult and to clone, and it had been shown at that time that a number of people said it was impossible to clone a mouse at that time. And we decided that a rabbit might be more closely related to livestock in the future, and potentially other species as well. And that's just led to a whole lot of breakthroughs in cloning technology, but also in the stem cell field as well. So we were really excited that this could be something that could have some great opportunities. And our first cloned rabbit was named Zeus. We were better at cloning rabbits than determining the sex of rabbits at a very young age, because Zeus turned out to be a female rabbit. But the name stuck, and we kept Zeus, and Zeus had a long life. And that's a common misconception about cloning, that cloning shortens the lifespan of animals. It truly does not shorten their lifespan. So beginnings as a graduate student, that took me all the way through stem cell biology in the future.
Yeah, it's always really fun to talk about the history of this field and the journey that different folks have taken in this field, because, in my lifetime, there's been such a tremendous evolution in the biology and the technology associated with stem cell biology. We have, of course, the reprogramming of iPSCs, which is really just about 15 years ago when that was first discovered. And now we have chemical reprogramming, and who knows what's going to happen in the next few years after that, right? I did want to sidetrack for a second and focus on where you've been for the last few decades. And that's actually the University of Georgia. And as somebody who grew up in the South, just down the road in Alabama, I'm always excited when we get to highlight work that's happening in your neck of the woods. And there's definitely more to UGA than just great football. It's an academic powerhouse, one of the best public universities in the US, and a leading producer of Rhodes Scholars as well. What is it about UGA, University of Georgia, that's made it a great environment for your work specifically, and it's kept you there for 2 decades?
That's a great question. And I grew up in the Midwest, went to the University of Illinois for an undergraduate degree, went out east and to Massachusetts for a doctorate degree. And we started up a company, STEMCELL Technology, there in Massachusetts. Never thought that I'd move to the South, but the environment here has been really conducive to my way of thinking about developing a technology like stem cells, and that it's great to be able to make breakthroughs in the lab, but if we're really ever going to make an impact in this field, we have to take that technology and commercialize it. And my opportunity here at University of Georgia that was afforded to me was where a role where I could play both as an academic advisor, scientific inventor at the University of Georgia, but also play in this other side of the sandbox on the corporate and industry development side. And as you mentioned in the introduction, I've been involved in a number of biotech startups, and recently Aruna Bio in the exosome field. And it's to me, the opportunity for me to do my best in the lab, but then also translate that technology into something that we hope we can be in clinical trials shortly. And that's holding true for Aruna Bio. We hope to be in clinical trials for stroke in the first half of next year. So all of those came together at the University of Georgia, plus the academic powerhouse. We've had a number of Rhodes Scholars, as you mentioned, but really top-notch individuals that have come through my lab, but at the undergraduate level, graduate level, and postdoc level that have made a major contribution. And the University of Georgia has a great foundation in biology, complex carbohydrate research, and so forth. So it is a great place to do research, but also gives me the freedom to do the type of things that I'd like to do to make this a reality for the industry and patients in the end.
Yeah, I mean, born and raised in New York City. I don't know much about the South, but I have to say that UGA also had a tremendous influence on me. When I first started working with pluripotent stem cells, embryonic stem cells, human, a great friend and colleague of mine, Scott Noggle, came from UGA in Brezogen and helped me derive the first human ES cell lines in New York. It was a really exciting time, finally being able to leverage human material to understand development. That was what it was for us. And the therapeutics, of course, that are just now starting to be realized. But it was also really steeped in controversy, right? It was a time where you really had to watch your back working with these cells. Brezogen ultimately generated multiple lines that were qualified for registration with the NIH, as I alluded to there in the intro. But, then iPS cells came, and it didn't seem like the urgency was there, and the attention maybe lapsed a bit. What do you think will come of those tremendous, gargantuan, monumental efforts and expense that was made by Brezogen, other collectives like it? Do you think that those and other lines will find widespread use, either academically or therapeutically? And if not, is there anything from that whole process, that whole rigmarole of interfacing with the government and academia that can serve as kind of guideposts for our current and future efforts to really bring these products to market clinically?
Great set of questions there, and that's a topic that I am really very opinionated on. I think the field of iPS stem cell technologies in general has been advanced tremendously, first by embryonic stem cell field. And Scott Noggle is somebody that came through the University of Georgia and then at Brezogen, so I know Scott. And the great people that started at Brezogen, I was involved in that group as well. And so we're, the things that work for iPS cells that are going into the clinics also are really standing on the shoulders of embryonic stem cell technology. And those are techniques that we developed very early on. I helped hire some of the first scientists that developed that technology at Brezogen, and today they continue to work not only in embryonic stem cells but also iPS cells. So I really see embryonic stem cells playing a major role going forward, because we know how they work. They're a gold standard for developing a stem cell that will develop into neural stem cells and can be used for treatment of Parkinson's. And even today, there's still a lot of interest in taking embryonic stem cells into the clinics as well. So I think there's no question that embryonic stem cells has played a major role in the development of iPS cells, but standing alone, I think they also have great opportunities for therapeutic options. And I think the world has learned that we're not deriving new embryonic stem cells every day. It's not the thing that everybody was fearful of many years ago that this would happen. We're really placing a lot of effort and understanding around what's the best techniques to use with a certain number of cell lines. And I think that's held true. There's some real gold standards in embryonic stem cells that people go back and use when they want to compare and develop something new for iPS cells. So all kinds of things that I can really describe that where embryonic stem cells have played a very important role.
Yeah, and even though human embryonic stem cells really put human stem cell research on the map and in a lot of ways into the public eye, it's no secret, like what we're talking about, the advent of iPSCs really changed the game and changed stem cell biology forever. There's an entire generation of stem cell trainees, like myself included, that rarely get the chance to actually work with human embryonic stem cells, given the ease of derivation of iPSCs and the fact that they can avoid some of these ethical controversies, associated with embryonic stem cells. And so, as a pioneer in human embryonic stem cell research and embryonic stem cell research, what do you think about the field kind of viewing these two cell types as quote unquote equivalent? It's what we've kind of been taught in grad school in a lot of ways is that, ESCs, human embryonic stem cells, and human iPSCs are pretty much equivalent. They're relatively interchangeable for their downstream applications. And when it comes to the training side of things, do you think that working with embryonic stem cells should still be a requirement for trainees in stem cell biology? Because like I mentioned, a lot of us have never touched embryonic stem cells.
Well, I don't think that it's a requirement. I do believe that there are things that were a lot of literature has been built around human embryonic stem cells in combination with iPS cells. So I think it's something that, depending on the need and the type of study that's going on, I think it's really useful to go back and reference those old studies and have the history and the historical data around them that can be and that you could then go back and thaw out a human embryonic stem cell line like an H9 and bring that back and test it in parallel with some iPS cells. So I think it's very important to be able to do those types of studies. And I think there's just a role for them in particular studies, but not for everyone. And I don't think that anybody missed out greatly by not growing up human embryonic stem cell lines, but I do think they play today, and in the future, an important role.
Yeah, for one, I can say for myself that when Scott and I, mostly Scott, I should say, he was the expert. But when we were deriving these lines, there was a thing about it where you putting a blastocyst on the feeder layer, there's such a gravity to that, where you're looking at and the implications and all the controversy surrounding it. So I don't think it's at all a requirement, but that there was something about that experience that really made you feel how precious it was and how you really had to be on point and not diminish the potential of this tremendous opportunity. But here we are now, moving forward, right? The translational applications of these cells that were so steeped in controversy are myriad. I mean, coming more every day. One of the many lines of inquiry along the translational track of yours is extracellular vesicles, so-called exosomes, derived from neural stem cells to be used for treating neural injury. Do you think the application of a cell-free regenerative product that is stem cell derived will find an easier path to market from a kind of regulatory standpoint, I guess, if not other standpoints? What are the obstacles you think to bring those to market? How do they compare to cell products?
Well, once again, I think part of the answer to that is the paving of the regulatory process with stem cells in general. So the groups that at FDA that regulate stem cell therapies are also regulating exosome therapies. And many of the, and that allows us to build on the opportunities that were afforded stem cells in a therapeutic option. We really think that, to answer your question directly, exosomes are the answer. They don't form tumors. You don't have to worry about many of the issues around this, injecting a cell that may have other effects in the body. We equate it to really, instead of having the battleship, you have the fighters that go in directly and attack the disease that is in question. The exosomes are small and can pass the blood-brain barrier much easier than a cell type can. So for us in the neural field, ability to administer a drug intravenously or intranasally or some other very systemic route of administration versus a direct injection of a stem cell into the brain or central nervous system makes a whole lot of sense. It may not make sense for all stem cell therapies and comparing that to exosomes, but something that crosses the blood-brain barrier readily and with minimal side effects and can direct that therapy to the brain just has all kinds of opportunities, not only as a therapeutic on its own, but since they're a master communication system between cells, exosomes or extracellular vesicles can carry payloads such as small molecules, nucleic acids, also proteins can get into the exosome, loaded into it, and then co-administered into the patient and have a great effect, not only the exosome, but also the payload that they are delivering. And it's a really 1-2 combination that has multiple opportunities for therapeutic options in the future. We think the time is now. There are companies in the exosome field that are in clinical trials. We think it's we're just at the cusp of taking a stem cell exosome and getting into the clinics with them.
Yeah, I think it's a field in exosome biology with tremendous potential and has really taken off, especially on the translational side within the last decade or so. And speaking of translating some of that exosome work, you're actually the co-founder of a bunch of different startups in biotech, including Aruna Bio. Great name, by the way. It's focused on the development of a neural exosome delivery platform and a pipeline of novel neural exosome therapies for the treatment of different neurodegenerative diseases like ALS, Huntington's disease, as well as stroke. And like we talked about, this is a bit of a different approach to what a lot of the cell therapy focused treatments are doing in terms of treating neurodevelopmental disorders, instead using exosomes as opposed to the cells themselves. But speaking more broadly, when it comes to the biotech side of things, what really fuels your passion for being involved in biomedical startups? And I ask because a lot of our trainees, more and more of our trainees are considering the jump from academia into biotech. I think it's a bit of a shift in terms of, the interests and the passions for trainees. A lot of trainees these days, they see a lot of potential in biotech and a lot of translational potential as well, right? And it's nice to have the perspective of somebody who's really truly translational in their work. So tell us what you think about that.
I think there's only a few opportunities to replace yourself. And I mean that in the sense that we shouldn't be just training people to replace ourselves, because there's just that few limited options for training new academics to come in. People that have great passion for academia should stay in that area, but there are more and more trainees that really want to get connected with the industry. And we offer that opportunity because we have a startup in next door. And many of the students interact with the employees at Aruna. They can see what it's like to be in a small company. So having those, opportunities, internships, informal internships, I think is really important as we train students today. I used to think, well, we should focus on publications at some level, but my thoughts changed considerably as we think about it further. We need to train the next set of or generation of scientists, but we really need to have them fully equipped so that they can go out and get into the technology immediately. That means they need to understand patents. They need to understand what's important in developing a product. It's not just the bench science. It's the company as well, that the finances are so important. This technology costs so much to develop into a therapeutic. They have to understand that time is money, and you have to develop things quickly. And it's not something that you can take a lifetime to develop. It just is something that we need to develop it quickly in order to develop it for the industry, but also for the patients. We want to get something in there into patients quickly. And I think that's what drives a lot of trainees today, is that, it's interesting to ask certain questions about science, and stem cells allows that to happen. But also they want to get to a point where it can be used and translated into a therapeutic. And I think more and more people understand that that requires some kind of business activity, whether it's a startup or a license to a large company. And we afford students that opportunity here to see that side of the development of a technology and provide that opportunity for them. So it's really important. We do that in conjunction, University of Georgia with Georgia Tech and Emory in a major NSF funded grant that we have called CMAT. And that's a great opportunity to train students that have direct interaction with industry.
Yeah, I think it follows from a kind of sea change in our field that is reflective of, I think, the great progress. I remember when we were just starting, it was a lot of hope. And I mean, best case, you come up with something great, and then, like you said, you license it out. But now I guess with this newer generation, there's really an expectation. It's like we know these cells are going to work, and we've got to provide the deliverables. So it's exciting for me to see this sea change. I think a lot of people cast it as like, oh, because there's no room left in academia, there's a surfeit of postdocs and no academic positions. But the reality, I think, is that there's more opportunity, and not just in human therapeutics too, right? Because there's all these other applications of stem cells and the field, some that are, directly related to biomedicine, but some that aren't. You've been active in seeking practical applications of stem cells and cloning in livestock, human pig chimeras, and genetically modified pigs that enable xenotransplantation of vital organs are just some of the really mind-bending advances that have been made in recent years. How far do you think we can take these applications for biomedicine? Also, this whole idea of cultivating meat. So it seems like there's a whole world out there where stem cells are applicable. How far do you think we're going to take that?
I'm always amazed at the new things that come up and how we can use these technologies. And that's what really gets me really excited. I never thought in the beginning when we were cloning animals that there'd be applications beyond just the agricultural application. But it turned out to be the first applications were in the pharmaceutical area, and we developed a relationship with Genzyme to produce pharmaceuticals in the milk of goats and cattle. And that was done through a cloning process. We could genetically engineer a cell type like a fibroblast cell and then make transgenic animals through the cloning process. So that was a turn I never thought that we would take at the beginning, but it's now probably the best way to use the technology in the biomedical aspects. And now I see another 90-degree turn where some of these technologies are coming back to the food industry. Some of the most recent things that I've heard about is these animals that have been developed for transplantation that we've knocked out a number of the antigens, gal knockout animals that make them more receptive for transplantation. Well, now they may be more a better source of food for people with allergies to food and some of the reactions that happen to certain types. So I never anticipated something like that, but for me, that's the most exciting thing, is the unanticipated opportunities that come about with a base technology like cloning or stem cells or exosome therapy. So in the future, it's hard to predict. We think there's great opportunities for therapeutic options with stem cells, but more importantly, exosomes. But there might be something beyond exosomes that will come from this technology, and I'm certain that there will be. What it will be is another question.
Yeah, you never know what might be just around the corner, right? And I guess we'll get close to wrapping things up with another application, another large animal application. We actually read up on a collaboration between your group and the Zoo Atlanta, where you're actually preserving the skin cells and the reprogramming iPSCs potentially of a variety of different species for the downstream production of sperm and egg. Right? So this is for the preservation of endangered species, a really important concept. I feel like this is a pretty unique application of stem cell biology, like what we've been talking about. It's actually taken off across a lot of different places in the country and around the world too. I remember recently talking to Gene Loring in San Diego about this as well. So tell us about how this collaboration came about and where things stand right now.
The collaboration with Zoo Atlanta really came from a student's interest in my lab that's now a full professor at the University of Georgia, Frank West, Dr. Frank West, who initiated that collaboration with the zoo and allowed us to take samples and grow up the fibroblast cells that then we could reprogram. And now we're banking those cells. The interesting part of that is that, again, we don't know where that will end up, but there's great opportunities to take that technology and apply it to preservation of new species. But also, there's a lot of work now taking iPS cells and turning them into gametes. Dr. Chaz Easley here at the University of Georgia has developed with people at Emory an ability to derive gametes from human iPS cells and embryonic stem cells. Not all the way yet, but very close to getting something there. So thinking of that in fertility treatments, but also for testing compounds that may have an effect on the human reproductive system and gametes. So we can use, again, in unpredicted ways, technology that we've developed for animals and turn that into something that's relevant and useful for human applications. There's a number of groups out there interested in preserving animals, and we're losing more and more species every day. Without those banks, it'll never be possible to bring them back. So it's really important to bank those tissues and be able to develop them into stem cell lines. We at Advanced Technology, I can say we did something with cloning where we could take a cow egg, transplant it with a Gower cell that's another cloven hoof animal that's endangered, and be able to use an existing source of oocytes or eggs to turn that into a new cloned animal from an endangered species. So again, and there's all kinds of branches where this technology can take us and that will help endangered species and animals in general.
Yeah, I love the idea that all the crowing that was done about, and I'm not going to make the ethical argument. People have their position, and I respect it. But it's ironic to me that all the terrible things that were being done with stem cells and destruction of embryos, and here we are, decades later, sure, but it seems like stem cells are really going to save the world. I mean, that might be overstating it, but definitely contributing to, conservation, as you just described, and sustainability, maybe with the meats from stem cells. So I would like to reflect on that for just a minute before we move on. And to finish, we have to ask you a couple science peripheral questions. I got to modify the first. It says, if you were not a scientist, what would you be? But I think in this case, you do so many things. If you weren't a scientist, entrepreneur, inventor, CSO, etc., what's left? I don't know. What would you be?
Yeah, that's a interesting question. I think what I enjoy most out of science is answering questions, the curiosity, but also putting things together and building them. So when I think about that and what I do in my spare time, I love puzzles. If I could be a professional or modeler, that would be great. Not in the computer modeling sense, but putting models together. Or as a mason, we've got landscape around our house that needs walls, and I've built a few landscape walls around them. I love doing that. If I didn't have a bad back and I wasn't as old as I was, that may be something I'd do. But I love building things. I love to see how we can put pieces of things together to make something bigger. So in that sense, love to do anything like that in the future.
I feel you on that, Doc. I'm the same way. Puttering around, if it weren't for my ailing back, I'd be a lot more active, much to the chagrin of my wife with all my home projects that most of the time don't end so well. Finally, what is the best piece of advice you've ever been given, either professional or not?
Yeah, I think it's a question that I get asked often. And I think the simple one, the most straightforward one is, you can do well by doing good. And as we train our students today, think about the good you can do with the technology, but also being able to translate that into technologies that are useful. You can develop wealth beyond just monetary wealth, but a number of different ways in which you can really benefit and profit in your life from this. So not speaking just about monetary aspects, but doing well by doing good, I think is a great motto to follow. And I've tried to follow that in my career.
Well, you've certainly done a lot, and you've done very well at it. Thank you so much for sharing these thoughts and your views and your experience with us. I'm sure our listeners can benefit from the real deep knowledge you have and firsthand experience over the course of decades. So really appreciate it, Dr. Stice.
Well, thank you for having me on your podcast. And anytime I can be of help, let me know.
All right, everybody, that brings us to the end of this episode. Thanks so much for listening. Don't forget to subscribe to our newsletter at www.stemcellpodcast.com to get the show notes, including an episode summary and links to all the interview and Roundup papers. You can also reach out to us on Twitter at STEMCELL or via email at info@stemcellpodcast.com with feedback or to suggest guests. We'll be back with an episode in a couple of weeks. Until then, thanks again for listening.